Hello, my name is Dr. Dewan S. Raja. Today I will discuss about the histology of the lysosome. The lysosomes are membrane-bound spherical-shaped organelle. These are present in the cytoplasm. They are formed from the Golgi apparatus. They contain hydrolytic enzymes for intracellular digestion. So these all are enzymes. These are proteins. They are derived from the Golgi apparatus. These are abundant in cells with highly phagocytic activity, like that of macrophage and neutrophil. They digest microbes and materials. So they digest the bacteria or cell debris or also the organelles of the cell. Okay. So primary lysosome and secondary lysosome. Primary lysosome contain inactive acid hydrolases. These are just derived from the Golgi apparatus. This is the Golgi apparatus. This is the transphase of the Golgi apparatus. These are the primary lysosome that contain inactive acid hydrolases. Secondary lysosomes are formed by the fusion of the primary lysosome to the endosome. Here is the endosomes or phagosome. Phagosome contain large material, bacteria, debris, large debris. And endosome is just the invagination of the cell membrane. It is containing some receptor and it also contains small particles. So it is formed by the fusion of the primary lysosome to the endosome, phagosome, autophagosome. Autophagosome, that means some of the component of the cell, suppose mitochondria, is covered by the membrane that is derived from the smooth endoplasmic reticulum that will be acted upon by the lysosome. Hydrolase degrades the content of lysosome. Indigestible materials are retained as residual body. So we'll get something, it is sometimes some of the components after digestion may not be digested, so that remain as the residual body that containing indigestible material. So this is the primary lysosome. We may have secretion, hydrolytic enzyme into the spatial microenvironment by exocytosis. It may come in contact with endosome or also with the autophagosome to form the secondary lysosome, and there will be digestion will take place. Some the digestive product remain undigested. They may be accumulated in the cell as the residual body. Other part which is digested that will be utilized by the cell. Okay. So lysosome enzymes are formed from Golgi apparatus because Golgi apparatus is the site of post-translational modification of the proteins. So they begin in the rough endoplasmic reticulum that is injected into the Golgi apparatus. In the Golgi apparatus, there is post-translational modification of the protein taking place, and those protein will go to the lysosome. So the from the Golgi apparatus, we have three destination of the protein. Some protein will go to the lysosome, some protein will go outside the cell by exocytosis, some protein from the Golgi apparatus will be added to the cell membrane. So lysosomal membranes walls are unique. We have to remember that We have the lysosome. Its walls are unique and are resistant to hydrolytic enzymes. Enzymes are derived from the Golgi apparatus containing mannose 6-phosphate targeting signal. So that enzyme or protein that is getting this targeting signal, it will go to the lysosome. Other protein will go to the cell membrane or go out of the cell by exocytosis. And we have to remember that lysosome is rich with the enzymes, around 40 to 50 enzymes. They are membrane bound. So inside the lysosome, there will be low pH acidic environment, and it is separated from the cytoplasmic comparatively high pH cytoplasmic environment, separated by means of the membrane, that is special membrane that is resistant to the hydrolytic enzymes. Okay. Secondary lysosome often produces debris from the undigestible product, filled vacuole called residual bodies, also called ACE pigment, and lipofuscin granule. This is the lipofuscin granule. This is the cell body of the neuron. Neuron is a permanent cell. So residual body are a normal feature of the cell, aging seen in neuron, cardiac muscle, skeletal muscle, because these are all the permanent cells. So we'll get the lipofuscin granule there. Defective enzyme in the lysosome causes lysosomal storage diseases. Okay, so we'll go to find out the lysosomal storage diseases. So lipofuscin granule is not only present in the neuron, it is also present in other permanent cell like that of the skeletal muscle, cardiac muscle. Okay, so these are the list of lysosomal storage disease. This has been taken from Jankuria's basic histology, and we have to know that some of these diseases are more common in some particular ethnic group of people like Ashkenazi Jews, and these are incurable diseases. Nervous system, liver, spleen, skeletal nervous system, muscle are affected in these diseases like Gaucher disease, Tay-Sachs disease, eye cell disease, Hurler syndrome, Mac-Ardle syndrome. So skeletal muscle is affected in Mac-Ardle syndrome. Gaucher Tay-Sachs, the nerve is here affected, Tay-Sachs disease, Gaucher disease, liver spleen is damaged. Okay. Okay. So, what is the highlight of today's discussion? Membrane bound organelle. Membrane is very much special membrane of the lysosome so that the acid content of the lysosome may not go to the cytoplasm. The membrane prevent the enzyme to leaking out. Cellular digestion is very important. It may be a phagosome, maybe an endosome, or it may be a autophagosome. The, the mitochondria or other organelle part is also digested by the lysosome. This is seen in case of poor nutrition condition. Okay, and also it is a part of the normal cellular process. Excess organelle are going to be digested by autophagosome. There will be a covering around the organelle by means of the smooth endoplasmic reticulum. Suppose excess mitochondria will be separated out by a membrane from the smooth endoplasmic reticulum that will be acted upon by the lysosome, and it will be digested and it will be broken totally. Okay. So endosome, that is on the cell surface, that contain receptor that is internalized and forms a cavity called endosome that will unite with that of the primary lysosome to form the secondary lysosome like that. Phagosome, these are large particle, debris or microbes that is picked up from the cell by a membrane, okay, that will also come in contact with the primary lysosome and form the secondary lysosome like that of autophagosome. It may be an organelle of the cell itself. So some of those are digested by the lysosome. We'll get lipofuscin pigment or aging pigment that is present in the permanent cell like that of the neuron and also cardiac muscle, smooth muscle. We have a list of lysosomal storage disease. And that's all about the histology of the lysosome. If you have any question, please feel free to ask me. Please share the information with your friends and please support my channel. Please subscribe me and have a nice day. Bye now.